from the Northeast. I'm not used to saying howdy yet. It's true, so I get made fun of a lot. Well, we're going to present our last speaker, Gyhye. Gyhye Yoo. She received her bachelor's degree in 2003 from Korea University while working as an undergraduate research assistant. Just want to say something I wish I had gotten to. She worked in the Division of Life Science at A&M University of Science and Technology, then went straight to her master's in plant functional genomics at the same place she had worked at A&M University, where she was also a research assistant until graduation 2005. Following her master's degree, worked out in the field for about 6 years. As a research scientist at the Korea Institute of Science and Technology, where she studied the role of WNT adenine signaling in colon cancer, as well as hepatoprotective effects of vegetables against chronic alcohol-induced liver damage. In 2011, she decided to make the journey all the way here. And she's a PhD candidate now at A&M. Through this department, well, our department of this is hosted by Nutrition and Food Sciences. Here she is a research and teaching assistant and had the opportunity to travel this country more than probably most of us, going to Boston, Washington, D.C., and all the way to San Diego, and of course, you presented in Korea. She's been featured in and even head author on as many as 13 articles and has won a couple of awards for her presentations, posted presentations, and things like that. She has a history of interest in both biochemistry and cancer research, which has brought her here today to speak about the influence of estrogen signaling and on colitis and colon cancer. Join me in welcoming our final speaker.
Thank you. Thank you for your introduction. I'm so glad to present my data here in nutrition seminar. Today, I'd like to present my data about the colon cancer and estrogen signaling. In our lab, generally, we have studied about the role of estrogen signaling on colon cancer. Among them, I am focusing on the influence of estrogen signaling on colitis, inflammation, and in colitis-associated colon cancer. Why is colon cancer so important? According to the statistics in 2014, colorectal cancer is the third leading cancer in the United States and the third leading cause of estimated death by cancer in males and females. Colon cancer is a consequence of a genetic mutation such as APC and KRAS and 53. Those genes mediate cell proliferation apoptosis. So when the intestinal epithelial cells have a genetic mutation, so to lose the ability to control the cell growth, they develop to the aberrant cryptophosphatide, which is a pre-malignant lesion. So this ACF develops to the adenoma to the carcinoma. Generally, this process take at least 10 to 15 years. Most of the colon cancer is derived from the sporadic colon cancer, almost close to 75%. 50% of colon cancer derived from the family history about the genetic mutation. And colitis-associated colon cancer is only 1% of whole colon cancer. Of course, you already saw the data. Okay, colorectal cancer is very similar in the both sexes. However, statistics have shown the premenopausal women has a lower risk of colon cancer than age-matched men. After menopause, women has increased the risk of colon cancer similar to the men. And if the postmenopausal women receive the hormone replacement therapy, it reduced the colon cancer instance again. So you can see this data. So after the 5 years of hormone repair therapy, reduced almost significantly in the risk of colon cancer. Based on this data suggested the protective effect of estrogen signaling against the colon cancer. So our lab previous data have studied about the estrogen could modulate the protective effect, has a protective effect against the sporadic colon cancer. So here's our data. So in wild-type mice, estradiol, which is an active form of estrogen in human body, estradiol treatment suppressed the A-shape formation significantly. And if the mice lose the ER beta, the effect of estrogen signaling was disappeared. This data suggested that estradiol treatment suppressed the A-shape formation, which means suppressed colon cancer instance via the increase of apoptosis. So we found estrogen signaling or estradiol treatment has a protective property against the colon cancer. In my study, I used genistein as a phytoestrogen. Genistein is a predominant isoflavone isolated from soybean. Because it has a very similar structure to the estradiol, genistein can bind to the estrogen receptor and modulate the gene expression. Generally, genistein has a higher binding affinity to the ER beta. Also, genistein has been reported a variety of biological activities, such as antioxidant and anti-inflammation. But still, the role of genistein on colon cancer remains debatable. During my PhD degree, I have 3 different topics. The first one is examining the role of novel phytoestrogen on colony epithelial cells. The second one is to investigate the effect of estrogen signaling on inflammation. And third one is to examine the role of 53 in estrogen receptor-dependent suppression of colon carcinogenesis. Due to the limited time today, I skipped the first part, and I just focus on the inflammation and the 53 part. I have 3 subtopics about inflammation. The first one is effect of estrogen estradiol or genistein on colony epithelials induced by interleukin-6. Second one is to investigate the effect of estrogen signaling on chronic inflammation. The third one is the effect of estrogen signaling on colitis-associated colon cancer. So what is the role of estrogen signaling on inflammation? Generally, chronic inflammation is positively correlated to the variety of cancer risk. Inflammatory bowel disease, IBD, is a chronic inflammatory condition in the gastrointestinal tract. There are 2 major forms of IBD. One is Crohn's disease, the other is ulcerative colitis. Generally, Crohn's disease occurs in anywhere in the GI tract, while the ulcerative colitis occurs in only colon and erectile area. Because IBD is associated with the aberrant T cell activation, Crohn's disease is associated with the T helper and T helper 17 activation, while the ulcerative colitis is linked to the T helper 2 activation. In spite of their distinct features of 2 different forms of IBD, both of them has correlated the colon cancer risk. So you can find here the colon cancer risk in IBD patients and almost close to 3 times higher than the healthy individuals. The role of estrogen signaling on immune system are very variable. For example, estradiol could function as a neurotransmitter in brain. Sometimes it can change the gene expression related to immune system. So depending on the status or tissues, it can modulate it can support or suppress the immune response. Furthermore, estradiol could suppress the inflammatory markers such as NF-kappa B or COX-2 and in the cancer cells or immune cells. Still, the role of estrogen signaling in colonic inflammation is not fully understood. So there are a lot of immune inflammatory markers, including cytokines, but I am interested in the interleukin-6, IL-6. IL-6 is a pro-inflammatory cytokine, which mediates the activation of TLP2 and TLP17 cells. When it binds to estrogen receptor, it initiates a cellular signaling cascade such as MET kinase, STAT3, or PI3 kinase signaling. If cells lose the IL-6 signaling, it impairs the tumorigenesis in breast epithelial cells. The relationship between the between the estrogen signaling and IL-6 signaling remains unclear. Only some reports have reported the estrogen signaling could modulate the IL-6 gene expression by inhibition of NF-kappa B, and the activation of MET kinase, PI3 kinase modulated the estrogen signaling effect on mammalian epithelial and breast cancer cell lines. Because those MET kinase and PI3 kinase is downstream of the IL-6, so IL-6 signaling could influence on the estrogen signaling as well. In first part, I have examined the effect of IL-6 on colonic epithelial cells. My hypothesis was IL-6 treatment will change the cell growth, apoptosis, and gene expression in colonic epithelial cells. And the effect of estradiol and genistein will inhibit the IL-6 effect via regulation of downstream of IL-6 signaling. I used young adult mouse colonocyte, YMCs, as a colonic epithelial cell model. Generally, it is really hard to maintain the colonic epithelial cells in vitro. So one group has developed this cell line for the in vitro study. So this is YMCs derived from the fertilized oocyte by mating BL10 and TBACA mice. Temperature-sensitive SB40 gene was introduced into the fertilized oocyte to initiate the conditionally immortalized colony epithelial cells. So YMCs grows continuously at 33 degrees, which is a non-permissive condition, while this growth slows down at 39, which is a non-permissive condition. It's very similar to the colony epithelial cells. So in vitro study, I treated the IL-6, estradiol, and genistein for 72 hours in YMCs, and then estimated the cell growth here, because the cell growth is a primary indicator of physiological change of cells. So I found estradiol and genistein suppress the cell growth, and then IL-6 treatment increased the cell growth significantly. So, and then IL-6, estradiol or genistein treatment suppressed the cell growth increased by IL-6. This data showed, okay, estradiol or genistein could inhibit the IL-6 effect in YMCs. Next, I measured apoptosis and cell cycle arrest because apoptosis and cell cycle distribution is a primary ways to control the cell growth. So you can see IL-6 treatment decrease apoptosis here, while the estradiol and genistein slightly recovered the apoptosis in YMCs. Unfortunately, I did not find any difference in the cell cycle arrest. So I think any treatment have influence on the cell cycle distribution. But this data indicates IL-6 increase the cell growth via suppression of apoptosis. And estradiol and genistein suppress the cell growth via increased apoptosis as well. Next, I measured the RNA expression because estrogen signaling and IL-6 signaling always control the gene expression. So I measured ER alpha and ER beta. So, there are two different major forms of estrogen receptor in body. So one is ER alpha, the other is ER beta. The ER alpha is associated with the cell proliferation, while the ER beta is linked to the apoptosis. So you can see and actually the previous data from our lab have demonstrated the ER alpha was increased and ER beta was decreased during the colon carcinogenesis. That's why I measured the estrogen receptor expression. IL-6 treatment increased the ER alpha expression and they decreased the ratio of ER beta to alpha. However, estradiol suppressed the increase of ER alpha expression, and genistein suppressed the ER alpha expression as well. Of course, I did not find any change of the ratio of ERs, but still, this means IL-6 effect on the YMCs was inhibited by estradiol and genistein. Next, I measured the CCND1 and PCNA because those genes are markers for the cell proliferation. So here you can see CCND1 and PCNA was increased by IL-6 treatment. Especially CCND1 is a direct target of ERs. So generally, ER alpha increased the CCND1, while ER beta decreased the suppressed it. So here, the increase of CCND1 by IL-6 indicates actually confirm the previous data about the increase the ER alpha expression by IL-6. And estradiol and genistein suppress the CCND1 expression. So these data suggested, okay, estradiol and genistein could suppress the cell proliferation in the cell line. Actually, it's opposite way to the IL-6 effect. The interesting part was IL-6. Most article has been reported positive feedback loop of IL-6 signaling, which means IL-6 generally increased IL-6 expression. But here in this study, I found IL-6 treatment to decrease the IL-6 expression. And I searched a lot of articles. I found some cases with the epithelial cell line, there is a negative feedback loop of IL-6 signaling. So because the YMC is not malignant cell, I think negative feedback loop of IL-6 could be activated in this cell line. And estradiol, genistein still decrease the IL-6 expression as well. In summary, I found the IL-6 elevates the cell proliferation in YMCs. Actually, this is the first data about IL-6 on colony epithelial cells, because most of articles has been reported of a lot of IL-6 signaling under the inflammation, not colony epithelial. And the part is this is the first in vitro data about IL-6. And I found the estradiol and adenosine suppress the IL-6 induced cell growth via elevation of apoptosis and regulation of gene expression. Next step, I think I need to demonstrate which downstream targets is modulated by estrogen signaling. So I plan to use the kinase inhibitors of IL-6 signaling, and then I'm gonna measure the phosphorylation of kinase using the Western blot. In second part, I have examined the effect of estradiol or genistein against the chronic inflammation. My hypothesis was estradiol or genistein will inhibit the inflammatory response in the colon against the chronic inflammation. The protective effect of estrogen signaling will alter the TLP17 and IL-6 signaling, because I already saw the estrogen signaling could suppress the IL-6 signaling in vitro study. So I would like to measure the TLP17 and IL-6 signaling here. I used the Plexus wild-type mice with the TMBS. TMBS is 2,4,6-trinitrobenzenic sulfonic acid, which is a pro-inflammatory agent. Actually, it is a kind of a heptinizing agent, so it can bind to the protein in the cell membrane, causing the immune response. So the treatment, the immune response induced by TMBS is very similar to the Crohn's disease, which means TMBS treatment induces a T helper 1 and T helper 17 activation. You can see the colon tissues here. The TMBS treatment to destroy the colonic crypt area and then increase the infiltration of immune cells in the rhaminophobia area. So first of all, we removed the ovary. We call the ovarectomization, OVX, to control the estrogen level in mice. And then we implanted the estradiol or cholesterol pellet and started the estrogen-free diet or genistein diet. So we have three groups. Control group has a cholesterol pellet with the estrogen-free diet. Estradiol group has a estradiol pellet with estrogen-free diet. And the last one is genistein group. Genistein group has a cholesterol pellet with the genistein diet. 2 weeks after the OBX, I smeared 1% of TMBS on the back skin of the mice for pre-sensitization, which means induce the primary immune response at the first time, because generally T cell activation is a secondary immune response. So, and then a week later, I injected 5 times of TMBS through the anus. Mice were sacrificed 2 weeks after the last injection of TMBS. Here's the body weight change. So you can see estradiol and genistein has a lower body weight compared to the control, because generally loss of the estrogen increase appetite, causing the mice eat more. And I measure the uterus because I want to sure the estrogen level is enough for the mice to protect the colon. So here you can see uterus weight was increased by estradiol or genistein. And then I measure the colon length and colon weight, because under the chronic inflammation, this is a primary indicator of colon status. So you can find the estradiol treatment to suppress the colon length and increase slightly in colon weight, which indicates estradiol has altered the change of the status of colon against chronic inflammation. Next, I measured inflammation and injury scores in the tissue. Actually, this data was analyzed by certified pathologist Dr. Weeks. Inflammation score means how much immune cell infiltration occurs in the tissue. The injury scores refers to the how how much damage of colony crypts occurs in the tissue. So you can see estradiol group did not suppress the inflammation, but slightly decreased the injury scores here. Surprisingly, genistein treatment increased the inflammation injuries too. So I think the genistein has a totally different effect of estradiol here. Next, I measure the cell proliferation in the crypt. Here's the crypt structure. So you can see that the whole crypt. So I divide the three different area in the crypt. Most of cell proliferation found in the bottom area because stem cells in the bottom area. So you can see most of the cell proliferative cells found in the bottom area, and estradiol increase the cell proliferation. Actually, this data was really opposite to my expectation, because we already know the estradiol suppressed the cell proliferation in sporadic colon cancer model, but this was totally opposite. So it was not my expectation actually, but still it's very critical. And even though estradiol treatment increased the total cell numbers per crypt. So these data indicate estradiol increased the cell proliferation, not suppress the cell proliferation here. Then I measure the RNA expression as well. So your alpha, beta, CCND1, PCNA, and IL-6. So here you can see estradiol and genistein increased. Of course, there is no significant data because I found the mRNA expression was really variable depending on individual mice. So I cannot get good significant data, but still you can see the tendency. So estradiol and genistein increase the alpha, beta expression, but the genistein only increase the ratio of ER beta to alpha. So it indicates genistein, it's kind of a higher binding affinity to ER beta. So it's that's why the ratio of ER beta to alpha increased only genistein. And I measure the CCND1 and PCNA. Those are the marker for the cell proliferation, as I mentioned. So here, estradiol and genistein increase the CCND1 and PCNA expression as well. Of course, the estradiol and genistein suppress the IL-6 expression similar to the in vitro study. So at least I found estradiol data is very consistent because this estradiol increase the cell proliferation markers and as well as the cell proliferation itself. So I would like to measure the cytokine to estimate the immune response here, because this is a chronic inflammation model. So I measure the IL-10, GM-CSF, and MIP-1 alpha at first. The IL-10 is an anti-inflammatory cytokine. So if IL-10 increases, means less inflammation. And those are the macrophage markers. So you can see those three cytokines were not changed in any treatment. And I move on to the cytokines related to the T cells. So as I mentioned, TMBS treatment similar to the Crohn's disease. And Crohn's disease is associated with the activation of T helper 1 and T helper 17. So I measured T helper 17 and T helper 1 cytokines here. So you can see left side is a T helper 17 cell cytokines. So IL-6 and IL-17 and CP1. So those three cytokines was increased by estradiol, while the cytokines related to the T helper 1 was decreased here by estradiol. It indicates, okay, estradiol modulate to the T helper cell cytokines, but cytokines, estradiol increased the cytokines related to the T helper 17, while decrease the cytokines related to the TLP1. In summary, I found estradiol induced the cell proliferation during chronic inflammation via increase of cytokine for TLP17 and decrease of cytokine for TLP1. Generally, TLP1 is associated with the removal of damaged cells via necrosis and apoptosis, while TLP17 is associated with the recovery of damaged tissues via cell proliferation. So my data suggested estradiol induced the recovery of damaged tissues via modulation of T helper cells here. Genistein exacerbated inflammation and injuries with no effect on immune response. This data suggested actually genistein is major effect of genistein is increase of apoptosis via ER beta. So I found that genistein increased the ratio of ER beta to alpha. So which means genistein itself has induced apoptosis here in this against the chronic inflammation. It indicates why I found more damaged tissues here, because it's more the genistein focused on the apoptosis. In further study, I would like to estimate the TLP1 and TLP17 cell population in the tissues. So I just I did not decide yet, but which marker is best for my experiment. In third part, I have studied the effect of estrogen signaling on colitis-associated colon cancer. My hypothesis was estrogen signaling will suppress the ACF formation against the inflammation-associated colon carcinogenesis. So I used the black 6Y mice with the TMBS and AOM. I already explained what the TMBS is, and IOM is azoxymethane, which is a colon-specific carcinogen. Generally, it just increases the DNA mutation causing the ACF formation. So here's the ACF structure. So you can see the very thick and large cells here. So that's the aberrant cryptophoci, which is a pre-malignant region. First of all, we remove the ovary and then inject the 4 times of AOM and 2 times of TMBS. 6 weeks after the last injection of toxic molecules, mice were sacrificed. In body weight graph, I found still estradiol and genistein suppressed the body weight gain, and estradiol increased the uterus weight as well. But I did not find the significance in the colon length and colon weight. But here I counted the ACF numbers. At least I found at least 10 more ACF numbers per mice. But estradiol and genistein suppressed the ACF formation here. Unfortunately, I did not find any significant change here, the proliferation or apoptosis in any treatment. So in summary, I found the AOM TMBS combination induced ACF formation. Actually, there are only 2 or 3 articles has been published about this model. And of course, this is first data of the role of estrogen signaling against the AOM TMBS model. So I found the estradiol and genistein inhibited the ACF formation. But I did not find any cell proliferation apoptosis increased by estrogen signaling. So I think it's kind of my theory, but I'm not sure about that. But generally, normal condition estrogen signaling increase the apoptosis. Main point is apoptosis here. But under the chronic inflammation, in second part of inflammation study, I found the estrogen signaling induced the cell proliferation, right? So combined together, in my study, the ending point, I think it's kind of a transition status about the cell proliferation and apoptosis, because I just put in the inflammation and the carcinogenesis together, right? So still, that's why I did not find any difference in apoptosis and cell proliferation rate, even though I got the reduced A-shape formation by estradiol. Okay, let's move on to the third part. It's about the sporadic cancer, colon cancer. As I mentioned, 75% of colon cancer is derived from the sporadic cancer. So my hypothesis was p53 is a major mediator of the protective effect of estradiol in the suppression of sporadic colon carcinogenesis. p53 is a tumor suppressor gene, as you know. It mediates the cell cycle arrest and apoptosis. Generally, any type of stressor exists in the cells or tissues, p53 activates the DNA repair system and the removal of damaged cell via apoptosis or other pathways. So this is a very important key modulator to control the cell growth. Previous data from our laboratory suggested p53 could be a mediator of protective effect of estrogen signaling. So you can see here data. So this is YMC data, so in vitro study. So estradiol treatment to suppress the cell growth. But if the YMC has a mutated 53, the effect of estradiol was disappeared. Furthermore, estradiol treatment increased the 53 level and modulated the gene expression of 53 targets. This data suggested, okay, 53 could be could play an important role on the estrogen signaling. So we move on to the in vivo study, but unfortunately we cannot use a 53 knockout mice because there are two problems. One is 53 knockout mice has a variety of tumors at the same time in very young age. So we cannot focus on the colon cancer here. And then second problem is female 53 knockout mice is very rare. So we think it's a 53 loss increase the lethality before birth. So, but we need the female mice. So instead we use the 53 plus minus mice in previous experiment. AOM injection increased the ACF numbers here, but in the heterozygous mice, estradiol treatment suppressed the ACF formation, but still the effect was estradiol was decreased compared to the wild type. So which indicates 53 is required for the protective effect of estrogen signaling. In my study, I used the Gyhye Yoo specific 53 knockout mice. It is called TP53 IC knockout mice. These mice were developed using the CRE-LOX system. Here's a CRE-LOX system. So CRE is a DNA recombinase found in bacteriophage. This enzyme recognizes the LOX site in the DNA, and then this enzyme excites out between the LOX site. So based on this system, two different mutant mice line was developed. One is fluxed 53 mice, which have a LOX site in the intron of 53. So if generally these mice express the 53 normally and functionally, but problem is if there is a CRE enzyme existing in these mice, the first axon of 53 was excised out, causing the loss of 53. The second mutant mice line is Vilin CRE mice, which has a CRE enzyme under the Vilin promoter. And because Vilin is a gene expressed only in the epithelial cells of GI tract, which means CRE is expressed only in the GI tract. So by mating two different mutant mice, we developed a TP53 IC knockout mice, which has which express the 53 normally in most tissues except for the GI tract. So it's kind of conditionally knockout mice. This mutant mice has been reported in some articles already. So you can see there is no phenotypic change at all in the knockout mice. But if after injection of AOM, you can find more tumors in the knockout mice. So I used the TP53 IC knockout mice in my study with AOM. So I injected 6 times of AOM, and then 6 weeks after the last injection of AOM, mice were sacrificed. So body weight change were not different except for the wild type estradiol, but still uterus weight was increased by estradiol in knockout or wild type. Actually, this experiment was over 2 weeks ago, so I still working on it. So I did not complete all data, but at least I just found the visual tumors. I counted the visual tumors when I collected tissues. So there is volumes inside there. So you can see the slight volumes here. So basically, knockout mice has more tumors than the wild type, and estradiol suppressed the tumor formation. But still, the estradiol has impact on the knockout mice. So I just found a little tumors in the knockout estradiol group. So now I'm working on counting the ACF formation using the microscope, and then I plan to the BRDO internal assay for the self-proliferation and apoptosis rate. In conclusion, I investigated the effect of estrogen signaling on inflammation and sporadic colon carcinogenesis. I found the estradiol protects the colon against the inflammation by a modulation of immune response. But depending on the condition, the effect of estradiol was different. Sometimes it increased the proliferation, sometimes it increased the apoptosis. The effect of genistein was not conclusive because basically genistein effect focusing on the apoptosis, not self-proliferation. So sometimes it exacerbated the inflammation. In sporadic colon cancer model, I found the estradiol suppressed colon carcinogenesis in spite of 53 loss, but I not conclusive because it's incomplete data. I would like to thank you, my committee members, Dr. Allred, Dr. Jarkin, and Dr. Weeks and Dr. Tarkot. Actually, I would like to thank Dr. Jarkin because I borrowed a lot of equipment from his lab. And Dr. Weeks analyzed my tissues for inflammation injury scores. And Dr. Tarkot, he's here to listen to my talk because he's give a lot of advice to how to present to the public. And I would like to thank my lab members. They give a lot of help for my in vitro and in vivo study. And I would like to thank Ms. Calloway. She is a professional for the animal experiment, so she gave a lot of advice for the animal experiment design. Funding resources are from NIH and HCS. Thank you for your attention.
We'll open the floor up for any questions. The lights full speed.
Your last study, the 53 knockout mice,
Yes.
is the way they can confirm it's a GI specific knockout. It wasn't blocked to verify.
Actually, it's not. Actually, it's not always development of our lab. It's already reported. So we received the 2 different mice, and then we just the mating by develop this knockout mice using the mating. So before we started this experiment, we just confirm the genotyping.
So the genotype is the only way, but there's no way to know is that tissue specifically knockout.
We can do that, but maybe we just confirm the.
Because
Well-
I think you mentioned that the protein is still there, right? It's just no function or just no protein at all.
Actually, it's non-functional because the 53 is expressed, but
Yeah.
without first exon.
So if you wasn't blood cannot, because the protein is still there.
Yeah, but it's non-functional.
But it's still there. If you wasn't blood, you should still see that.
Yeah, depending on because if we just use the antibody for the first exon, I think we cannot recognize anything, but
Yes.
the other parts, yeah, it could be. Yeah.
The other way
Yeah.
to verify is knockout.
Well, we use the genotyping, so yeah, we did not think about that. Yeah.
We also characterize some of the downstream mediators that are P53 dependent. Those mice.
Well, yeah, it could
We
be,
have
but
those data.
So that was, I just want to know whether there's a way, except the genotyping, whether there's a way you know that tissue specific knockout. There's no P53 in Gyhye.
Yeah, so you can look, P53 is a transcription factor, and so you can look at P53 dependent genes that are knocked
Downstream.
down. So we did some of that to prove that it was significantly impaired. We'll get that in catch.
Yeah.
No, I just wanted to say, you said that genistein wasn't conclusive because it caused more inflammation because of apoptosis. Is that what you said?
yeah, because I did not confirm by theory using by any experiment, so I cannot be conclusive because if I just because I did not find any increase of the proliferation, and I'm now working on it about the apoptotic cells in the crypt. So I did not finish that data, so that's why I cannot conclude. Yeah.
So in your model, you treated with TMDS and in some cases in TMDS and AOM in another experiment.
Yes.
And you found that the estrogen was enhancing the Th17 cell phenotype in that particular case. It was promoting things like IL-17 and one or two other.
IL-6 and MCP1.
Was estrogen promoting those as well?
Actually, I did not find any articles about estrogen signaling could modulate that cytokine levels, especially in the colony epithelial.
No, I mean your data. What did you find?
I found the IL-6, IL-17, and MCP1 elevated by estradiol. So I think TLP17 activation or maybe more recruitment into laminophilia. So that's why I just do measure the T helper 1 and T helper cell population in the laminophilia area.
But you think you interpret your data to mean that the colonic mucosa might be more resilient, be repairing more rapidly after whatever TMDS induced
Chronic inflammation.
is occurring. Is that right?
Yes.
So the Th17 cell is a good guy potentially in this model because you're enhancing that signature.
Yes, I think so. That's actually that was not my expectation because in vitro study I think estrogen signaling suppressed IL-6 signaling to protect the colony epithelium, but in current I did not mention about Cameron, previous student in our lab, was a study about the acute inflammation by TMDS. At the time, she found the estradiol treatment suppressed the IL-6 signaling as well in acute inflammation. So at the first time, when I started this experiment, I expected maybe estradiol protect the colon because it prevent the damage of tissues. So I expect that, but according to my data, it's totally opposite. Estrogen signaling protects the colon via the increase of cell proliferation. I mean, increase of wound healing, not the prevent the damage.
Okay.
Yeah.
In the TNBS only study, how long after the last administration did you try?
2 weeks.
That's a long time after.
Yeah, that's why I think there is no change in the macrophage or anti-inflammatory cytokines, because, of course, at the first time I designed this experiment, I have think about which is the best termination point for the immune response, because generally in acute inflammation study, we just kill the mice after 5 days after of TNBS injection. So, yeah.
So I'm a little surprised that proliferation is still elevated.
Yeah, for me, yeah, it was really surprising me. It's very significant. So, yeah.
Can you comment about your findings with genistein?
Yes.
I'm just wondering, any hypotheses on why you found what you found? My understanding was that you chose genistein, correct me if I'm wrong, because it's like estradiol.
Yes, but generally, estradiol, it's an active form of estrogen in human body, has a higher binding affinity to the ER alpha. But it can bind to the ER beta as well. So generally, the ER beta is a predominant form of ERs in colony epithelium. So I think ER beta is a mediator of estrogen's protective effect of estrogen signaling in colony epithelium. But here, in this study, I don't know why, but the genistein still increased the ER beta expression. So that's why genistein increased the apoptosis instead of increase the cell proliferation. So that's why the genistein has no good beneficial effect on the wound healing. So that's why I found more inflammation, more injuries here. But in vitro study, I just make a inflammatory condition using the IL-6, right? So generally inflammation status has a lot of cytokines and inflammatory markers like NF-kappa B, COX-2, something like that. So it's a little bit complex status, actually, that's inflammation. But here I just make a artificial situation using the IL-6 only. So that's why genistein actually increase the hypothesis. That's why protective colony epithelial cells. But during the chronic inflammation, it's in vivo study. So that's why basically genistein has anti-inflammatory properties already. So in a number of cancer cells or epithelial cells. So that's why I think genistein exacerbated the inflammation here. Yeah.
In the TNBS study, how did you measure proliferation?
I just using the BRDU staining. So I injected the BRDU before I killed mice, and then I just staining the BRDU antibody. Yeah.
Any other questions for our PhD candidate? In that case, let's give